The third important component of periodontium is cementum. In the last video, we learned how periodontal ligament fibers stabilize the tooth in socket. But what provides anchorage to these periodontal ligament fibers? The answer is cementum. Cementum is the mineralized dental tissue covering the anatomic roots of the human teeth. It begins at the cemento-enamel junction and extends up to the apex. It is calcified and avascular. Cementum is light yellow in color and can be distinguished from enamel by its lack of luster and its darker hue. It is slightly lighter in color than dentin and is also less hard than dentin. Cementum thickness varies in different teeth and in the same teeth in different regions. It is thinnest at the cemento-enamel junction and thickest towards the apex and in furcation area. The apical foramen is surrounded by cementum. It is also thicker on the distal surface than the mesial surface, likely due to continuous functional forces from mesial drift over time. Coming to its composition, chemically, cementum contains 45 to 50% of inorganic material, that is less than that of enamel and dentin, and 50 to 55% of organic material and water. The inorganic portion consists mainly of calcium and phosphate in the form of hydroxyapatite. The major portion of the organic matrix of cementum is composed of type 1, that is 90%, and type 3, about 5% of collagen. Sharpie fibers are composed of mainly type 1 collagen. Type 3 collagen appears to coat the type 1 collagen of the Sharpie's fibers. The other types of collagen seen are type 5, 6, and 12. Apart from this, cementum also has non-collagenous proteins like osteopontin that regulates mineralization, bone cialo protein that promotes mineralization, and cementum attachment protein that is responsible for cell adhesion. It also contains proteoglycans like chondroitin sulfate, heparin sulfate, hyaluronate, and keratin sulfates, and growth factors like transforming growth factor beta, bone morphogenic proteins, fibroblast growth factors, and insulin like growth factors. These help in matrix formation, mineralization, and cell differentiation. Now, if we look at the structure, cementum is composed of fibers, cells, and ground substance. The fibers are of two types, intrinsic fibers and extrinsic fibers, that are Sharpie's fibers. The cells include cementoblasts, cementocytes, and cementoclasts. The ground substance consists of proteoglycans and glycoproteins that occupy the space between fibers and cells. Before we understand the different types of cementum, let's first see how cementum is formed. The internal and external enamel epithelium proliferate downward as a double-layered sheath of flat epithelial cells called Hertwig's epithelial root sheath. This sheath induces the dental papilla cells to differentiate into odontoblasts. These odontoblasts form the first layer of root predentin. This layer is structureless because the odontoblasts do not leave behind odontoblastic process, and thus it is known as the hyaline layer. Subsequently, Hertwig epithelial root sheath undergoes fragmentation, allowing the dentin to come in contact with the cells of the dental follicle. These cells differentiate into cementoblasts, which are responsible for cementum formation. Cementoblasts start secreting collagen fibers and ground substance. Initially, the fibers are parallel to the root surface. Later, they become continuous with the periodontal ligament fibers. The unmineralized matrix formed is called cementoid, which later undergoes mineralization, and cementum is deposited in a rhythmic manner. This results in the formation of incremental lines known as lines of Salter, which represents periods of activity and rest. The first formed layer of cementum is known as acellular cementum. If cementoblasts get entrapped within the matrix, they become cementocytes, and these are present in lacunae and helps in maintenance of cementum. leading to the formation of cellular cementum. Let's move on to its classification. Cementum is broadly classified based on the presence of cells. Acellular cementum does not contain cells, and cellular cementum contains entrapped cementoblasts called cementocytes. It is important to clearly understand the basic differences between these two. Acellular cementum does not contain cementocytes, is formed slowly, and is mainly found in the cervical third, with well mineralized Sharpie's fibers and closely spaced incremental lines. It is also known as primary cementum. In contrast, cellular cementum contains cementocytes, forms faster after acellular cementum, and is mainly seen in the apical and interradicular areas. It is called secondary cementum, has partially mineralized Sharpie's fibers, and shows widely spaced irregular incremental lines. Another classification of cementum by Schroeder is based on the type of fibers, intrinsic and extrinsic, and the presence or absence of cementocytes. First is acellular afibrillar cementum that contains neither cells nor fibers and is made up only of mineralized ground substance. It is mainly seen in the coronal region and does not have a significant role in attachment. Acellular extrinsic fiber cementum contains no cells but is rich in extrinsic fibers, that is, Sharpie's fibers. It is mainly found in the cervical third of the root and plays a major role in anchorage of the tooth. Next, cellular mixed stratified cementum contains both intrinsic and extrinsic fibers along with cementocytes. It is commonly seen in the apical and furcation areas and is involved in both attachment and adaptation. Last, the cellular intrinsic fiber cementum. It contains cementocytes and only intrinsic fibers with no extrinsic fibers. It is mainly found in area of repair and plays a role in regeneration rather than attachment. These intrinsic fibers are produced by cementoblasts. Lastly, intermediate cementum is a thin layer near the cemento-enamel junction that contains remnants of Hertwig's epithelial root sheath and has no major functional role. Next, let's look at how cementum relates to adjacent structure at its junctions. First, cemento-enamel junction. It is the junction where cementum meets enamel, and in 60 to 65% cementum overlaps enamel. In 30% they meet edge to edge, and in 5 to 10% they do not meet, leading to dentin exposure and sensitivity. Next is cementodentinal junction. It is the junction where cementum meets dentin near the apex. It is important in root canal treatment and is about 2 to 3 micrometer wide with intermingling of fibers that ensures attachment. Now let's move on to the function of the cementum. Cementum performs multiple important functions including anchorage, adaptation, repair, and protection. It provides anchorage by serving as the attachment site for periodontal ligament fibers. It plays a role in adaptation through continuous deposition, helping to compensate for occlusal wear. In addition, it contributes to repair by forming new cementum after resorption and offers protection by covering the root dentin. Now that we understand cementum and its function, how does all of this translate clinically? Cementum is relatively more resistant to resorption than bone. This is why during orthodontic tooth movement, bone resorbs more readily while cementum remains relatively stable. However, cementum can undergo resorption under certain conditions such as trauma or excessive occlusal forces. Once resorption stops, repair can occur by deposition of either acellular or cellular cementum. This repair can be of two types: anatomic repair, where the original root contour is restored, and functional repair, where the contour is not completely restored. Another important condition is ankylosis, where the tooth becomes fused directly to the bone due to loss of periodontal ligament. When cementum becomes exposed due to gingival recession, it can be affected by cemental caries. Also, in periodontal disease, the surface of cementum may undergo hypermineralization. Additionally, excessive deposition of cementum, known as hypercementosis, may occur due to occlusal stress or inflammation and can make extraction difficult. Finally, the incremental lines of cementum can be used in forensic age estimation. So this completes cementum. In the next video, we'll simplify classification of gingival and periodontal disease because the 2017 classification is quite vast and can feel a bit confusing. We'll break it down into a clear and structured approach so it becomes easier to understand and remember. Thanks for watching. Follow and subscribe to Gamodont for more simplified periodontics.